Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.
Okay, sorry about that. So we're back. Okay, I'm here with Tim Kram from Rommelag. We're going to be talking about automated visual particle inspection for hard-to-inspect containers. My first question is, what are hard-to-inspect containers?
Okay.
Hello?
Yeah, you got to talk into that thing. Yeah, there you go.
All right, if you can't hear me in the back, please wave or something, because I sometimes get kind of quiet. In general, hard-to-inspect containers can be twofold. One, because the product is difficult to inspect because of its characteristics, or two, what we more work towards is containers themselves that present a problem. So opaque glass or blow-fill seal containers that are regular, or flexible bags where maybe pre-printed or other things have been done to them that interferes with the inspection process.
Okay, I got that. So why do we inspect stuff? Let's talk about that. Why do we use visual inspection? I mean, to me, with the new QBD and CQA, all the quality is supposed to be built in, and let's talk about that. So we visually inspect. What's up?
Well, ideally, we would not visually inspect, but getting more towards the Japanese model of building cars, if your process is perfect or very good, then you wouldn't have to inspect. Throughout history, though, we have found that there are defects in containers, and particularly we've taken the regulatory bodies have taken the position that on injectable products in general, you need to do a quality check at the end of the process to ensure that your product going out is essentially particle-free.
I get it with the, if you look at a glass vial, I imagine it's pretty straightforward what I'm supposed to be looking for, but obviously I'm missing half the story here. I mean, so, the answer is, what is it, blow-fill seal? I mean, what?
Oh.
Yeah, so what am I,
There are technologies, definitely you can start with your process, and if you start from a risk basis of having fewer particles, that definitely can improve your overall quality of your product. So at the end of the line, if you start with a lower particle load, if you don't have to clean the containers, if you do fewer steps, every step you take in the process adds risk. You have to evaluate that and see what kind of particles can be put into it. So you have your intrinsic particles that are, that come with the containers, come with the process, and extrinsic, so maybe in an environment around there. In general, it's kind of a given that your intrinsic particles, you try to improve your process, you minimize those, but everybody kind of assumes, or there's a consensus out there that you can't get all of those out. Now, extrinsic particles, I mean, a good human hairs, a bug or something like that, there's a very frowned upon, and you definitely have issues if you can let particles from outside your system into it. It's all based on process. So I mean, we'll be looking at blow-fill seal containers. Blow-fill seal containers have the advantage that we are creating them in the machine. You make them, you fill them, you seal them, and it's in a very short, controlled environment, so 12 seconds. If you compare that to a vial where you buy it, have it delivered, you store it, you have to clean it, depyrogenate it, put it into an open area again to fill it, and then seal it sometime after you fill it. All those steps and all that time adds risk to it. So it is looking at the overall process. But then plastic is not for everybody too. So
I would think the intrinsic problem, let's call it extrinsic. In other words, I would have thought we got a better handle on that. What's been your experience? It seemed to me we're in 2018.
Correct. From what I see, people starting with the same raw materials, the same empty containers, there's a great variety of final product quality. So we're not there yet. People do things, some people do it better, some people do it worse, or are not aware of where the issues are coming in. So that maybe there's still a necessary need to improve the the overall systems for particularly aseptic or processing. So
Yeah, I find it a little hard to believe when you go out here and there must be 10 or 12 people that sell environments to eliminate that very thing. No, I'm just
right. You're right. It's much better than it used to be. But I mean, if you look at, there was recently a study done on the number of isolators and closed wraps. So there's the 3 advanced aseptic processing technologies for fill finish: closed wraps, isolators, and blow-fill seal. Last year, more traditional aseptic lines went in without any barriers. So that outnumbered the closed wraps and the isolators and the blow-fill seal systems. So people are still sticking to the older, technology of just a clean room with people gowned up in bunny suits. And it's vastly agreed that the number one, the highest risk for contamination comes from people. And if you have people standing next to an open container, you are relatively taking a big chance compared to having some type of barrier or something in between you and the product.
So the same. I mean, what is that? What's the barrier? I mean, not zero barrier, but what's the problem? I mean, if I can do something that's going to eliminate that, what's the barrier to doing that?
My personal opinion is it's perception of flexibility and cost. So the cheapest way to do it is a traditional clean room. And if you have lots of changeovers or things like that, people think you get away, if you put a barrier around there, you're going to impinge on that ability to be flexible.
Oh,
so
and they value that against the recall, which can get a little pricey,
exactly.
Right? Okay. All right. Hey, while we're on the topic then, let me just say, so what are the standards that, for particle inspection? Talk to me. I mean, I thought, so in other words, is there a standard requirement or what's the deal?
At that point, we're looking at the standard is human inspection currently. So whatever you have a set piece where you're inspecting, and what people can find under a controlled test is you set your standard. Many people continue with that, so they're doing inspection manually. That's perfectly accepted. There are a lot of automated systems out there, and the automated systems are validated against the human inspection. What people can see depends on the product, the container, the design, optically, the lighting, things like that. There are some standards there too as far as how you're supposed to set up the inspection. But I think in glass, the goal now is sub 100 micron, finding particles in that side for clear liquid. On plastic containers, which is primarily what we work on, we're in the 200, 250 micron to find. So that's the current technology.
Well, it just seems to me that, the visual inspection part, because everybody's talking about continuous this and everything's got to be, it's got to be a big speed bump in the process, doesn't it?
Correct. So the inspection requirements are becoming more stringent, and then the automation level is also coming faster, so your equipment has to do the inspection as well or better, but also faster. And people, if you're still using manual inspection, you have to have more people, because if the machine spits out more containers, that inspection is still at a fixed rate. So the only way to expand that is to have more people, and more cost. And I think the FDA has expressed what they would like to see is, again, like you noted earlier, was improve the whole process. So get to the point where you're not having recalls, you're not having particles found in general. So if you have particles in your process, go look at your process and eliminate those particles in the first place. Don't use inspection as a fail-safe. You shouldn't find anything in inspection. But
exactly. I mean, that's like developing a tablet where you depend on the weight control monitoring system to take out all the bad stuff.
Yeah.
It's called excursion, if I'm not mistaken.
Okay.
So anyway, so people still depend on that. So, all right. So Tim, let's talk about it. So, what approach should I take? I mean, if I was getting into this, and I think one of the things that we used to do was that a lot of the injectables were real quick and dirty. I don't mean that, sorry.
Yeah.
But that, it was like, we did that as a reference standard for a BE study. I mean, we're not talking about stuff like that. What I'm talking about, stuff that the intention is to go beyond just a basic scientific approach. We're coming up with a product. So from square one, today I was going to say, okay, I want to make this sterile product, and I want to avail myself of quality. So what steps do you take? I'm going to start from the formulation and work forward from there.
Okay. I'm focused more on the fill finish portion of it. So,
that's good.
If you're, particularly for aseptic products, in most cases what you're looking at is everything past the aseptic filter. So you do have a final filtration that let's say the higher value products, a lot of products now are biotech and they can't go through filtration.
Right, okay.
They have a larger challenge in this, but
okay, let's go with that then.
yeah, the final steps, having a clean container prepared and ready to go in. If we're looking at a lot of products now are going into larger volumes, so IV type flexible bags, ready to basically give to the patient in the hospital rather than mixing it in a hospital setting. These type of products, you're totally dependent on your incoming materials and then your ability to take them and clean them. And with many materials, that can be difficult. I guess it. Getting back to blow-fill seal, maybe more my area. Here.
Well, that's why I was hoping it was going to.
So the traditional methods do have built-in issues, and then the blow-fill seal, where we create the container, we fill it, and in some cases, and that's all done in 3.5 seconds, and we discharge it. So by making a container that never has any exposure to external contamination, we don't find endotoxins or spores on the inside of the container because it's physically impossible or very rarely for those materials to get inside of the container. And then what follows there is that once you've sealed the container and get it out, then you're relatively it's a lower risk process than what you find in traditional manufacturing.
So what percentage of people, it seemed to me intuitive that this would be something you would pursue, considering that we're trying to get rid of the acceptable limit of black specks in the API. So.
Sure. The use of that technology within the U.S. is limited. It is better accepted outside of the U.S. and Europe and Asia. I think there's a traditional reluctance to use new technologies in the U.S. We're using glass and stopper, and that's the easiest way to move forward. You also install base of equipment and facilities, and that's a practical, reason to go forward with that. And then there's always the concern with actual compatibility with the materials. Glass is relatively inert and has a high barrier, whereas plastic is compatibility issues with the material itself, and also low barriers to, let's say, gas to oxygen. So secondary steps have to be taken then to protect the product.
I mean, compatibility studies are done all the time.
All the time, yeah.
All right. I mean, contact studies and multiple, and in injectables, the same thing. So
Correct.
How many variations do we have to look at then?
In some respects it's easier because you have a monolayer container. You only have one product contact. You don't have glass, silicone, a rubber stopper, any of those materials involved. You basically just have to have one polyethylene or polypropylene material that you're in contact with.
Right.
So that's the primary contact level. So in some respects it's more straightforward, but when you throw in permeability on adhesives and things like that, and the external packaging, secondary packaging that can affect your product, there are concerns there. So it's not you have to run through the whole process of extractable leachables and testing for them.
So this eliminates that aspect.
Well, it's different than glass, but
Yeah.
in some ways it's better.
Okay, so it limits my variability. The question really comes back to is there anything that I would not encounter? I mean, imagine if I was going to go through a fill and seal, blow fill seal, I would have to assess that, my product's going to be relatively, is it going to have to be very, very heat stable, or is it, you mentioned 3.5 seconds, that's the dwell
Sure.
time?
Done a lot of work with vaccines and biological products, and what we found there is most products, not all, are, their temperature sensitivity is an elevated temperature for, an elongated hours or days. Blow fill seal imparts a little bit of heat for about 15 minutes. So typically you're having a raise of 4 or 5 degrees Celsius, and then you can bring it back to room temperature relatively quick. So it does impart some heat. It's a warm process. Let's say it that way.
And that heat comes in where when you form
the container is still, when you extrude the plastic, it goes into the mold, and when the mold closes on it, you're forming the container, and so that's the bulk of the heat is removed at that time. But by that second and a half or 2 seconds later, when you introduce the product, there is still some residual heat, particularly on it. Plastic's an insulator, so you're cooling it from the outside, so the inside of the container still has some warmth to it. So that will go into the product and warm it up. For particularly heat-sensitive products, what we do is we'll bring down the temperature of the product prior to filling, maybe to 4 or 5 degrees Celsius. For a 2 milliliter product, we'll go up maybe 8 to 10 degrees Celsius on top.
What sort of ranges on that volume are we talking about then? Minimal to maximum?
Most of the products we're talking about here are in and around 1 milliliter. We can, once you go to 5 or 10 milliliters, there's almost no impact from the heat. It's 2 or 3
Agreed.
degrees. It's not very much.
Okay. So is there a low end, high end? What is there?
For
If
filling,
I was going to go down that route to blow fill seal, what's
we've
the
gone down to
technical limits?
50 microliters on the low end, and on the high end we do liter bottles. So it's really kind of everything in between. A normal size fill range on the small end is 100 microliters.
Okay,
So
and is
2 or
that
3 drops.
complete recovery then? Or is there any residual hanging there or what?
Depends on more if it's water. Actually, water is not great, but if you have an API in there or some buffers, typically, your repeatability on getting all that product out is fairly good. You'll lose a little bit of it. Water itself actually doesn't come out that well because it sticks to the side of the containers. But
Understood.
once you put other materials in there, it flows pretty well. It doesn't. The inside of the container is fairly hydrophobic, so as long as you don't have a high surface tension on the product, it'll flow pretty well. Gels and creams are a little different. So
Gels and creams.
gels and creams will leave some residual.
So other than sterile, people are doing the topicals and
Yeah, we do some sterile suspensions, sterile gels for ophthalmics, so we can do those too. So
how about dry material? Dry
we
fill?
don't do dry at all.
Don't do
So,
dry, right?
no.
No, I was just thinking because
Yeah.
a lot of the oncology,
The
they go down that
the
road.
dosing is very difficult. So, getting it in there, but getting it in there accurately when in the small environment that we have is very difficult. So,
Okay, all right. So typically, biologics. What's been your experience then? Is it solutions or biological
typically
suspensions?
suspensions.
Yeah.
Suspensions mostly. Yes. So, getting them in there is no problem. Compatibility with the resin.
Okay.
Adsorption into the plastic is kind of the death nail for that type of product.
Right.
So if you can't deal with the materials we have available, then if you can't deal with the materials we have available, then it's not a good choice. So
What about uniformity, stuff like that, dose to dose? What's it been?
we're using a time pressure dosing system, so it's similar to what you get in those others. We also have an option to use peristaltic pumps. We use a Bosch system, so that's kind of outside of the packaging itself. So it's similar to what you find in other systems.
Okay, so can this process be contained as it is, or do you have to have it within the sterile area, or what?
The requirements there, you have to have the machine within a ISO 8, so 100,000 room. So,
Okay.
And then that the machine itself creates its ISO 5 or Class A filling environment and controls that. And if you interfere with it, then it shuts itself down and makes you clean the machine and steam it again before you can start back up. So it's fairly, excursions into that area are pretty much cause you to restart the machine and re-clean it. So in that respect, it's fairly robust.
How much flexibility do I have on the material that makes up the vial?
Anything that can go through extrusion system, blow molding extrusion system.
Such as?
Polyethylene, polypropylene, co-extruded materials. So PETs do a little bit in that for some cosmeceutical products. So
Okay.
it's prettier. And some of the COC, COP materials now are into being produced in materials that we can use. And one of the challenges there was always it was a very tough material, and we couldn't, you couldn't do a twist off with it. It was too brittle. But now they've come up with some softer formulations that still give you the good chemical compatibility and barriers that you want, but they're softer and they're more. We can use them within the blow-fill seal system.
Is there multi-compartment aspects to this? In other words, of having
We
compatibilities?
quite haven't figured that one out, so no co-mixing of liquids. So we could do something similar like the syringes where they have a valve or something like that, but we can't really have two, like the film barriers which
Right.
rip apart. That's not possible. So
Okay.
because we fully seal together.
Yeah, because those things those from a formulation standpoint. It's always nice to know that I can ultimately fix it with a packaging issue. But ultimately, so you're telling me no, huh? No help there.
If we figure it out, there's lots of people waiting to do that. So
Okay, good. I hear you. So what about identification, labeling, all this rest of the stuff? How easy is all that to incorporate here?
Secondary packaging is very similar to.
How about on the package itself?
It's no problem. So
Okay, and where is that done?
Outside of the clean room. So
Okay.
it'll be full CNC, normally controlled non-classified areas, typical, just clean.
Okay.
So
So what other applications have you seen? I mean, this thing sounds like it could be pretty much anything.
the traditional applications are in ophthalmics and inhalation, nebulized inhalation drugs, injectables, local anesthetics. More we're seeing IV type products worldwide.
Yeah.
If you go to Germany, you'll get a bottle instead of a bag. That type of application. And lots of interest from people that are having compatibility issues with glass. So if you're delaminating or you're pulling metal ions from the glass, then plastic looks better to them. So
In this environment of pediatric dosage forms and stuff like that, have you seen this come up as an opportunity for this area?
yep. Again, in the traditional ones in inhalation and ophthalmics, yes, definitely.
What about suspensions, oral liquids, stuff like this?
Yeah, where you can pre-measure doses and things like that. That's a popular application.
So this is used in the unit dose
Correct.
approach?
Unit dose.
So it's comparable as far as cost and everything in that case?
Yeah.
Excellent.
A lot of that is your capacity. So it's a high capacity, low cost. So if you merely want a few million, sometimes that is not worthwhile for the installation. But if you need 20 or 50 million, then it becomes more viable. People are typically using contract manufacturing for smaller quantities.
Okay, well, while we're talking about quantities, how about changeover? How much flexibility do I have? Is this a one size?
The bigger the machine, the less flexible, the more dedicated typically. That's how people run them. So if you have a machine that's, at 600 vials a minute, then you keep it running the same thing. When you drop down to, 1/5 that, then you have a machine that does lots of different sizes, and it's easy to change over. So
I got you. So if I was looking to, if I was a contract manufacturer and I wanted to be able to have something, I like the concept because it gives me a lot of quality assurance aspects to it. But how about my flexibility because I want to deal with my customers? So what do you recommend in that particular case?
it really depends on what you want to get. Or produce. It's easy to do something that's very flexible in a family of containers or in a size range. Maybe 135's, 10 milliliter, or 10, 20, 50 milliliter, or 100, 250, 500 milliliter. So those are kind of, when you get synergies between the container sizes, then it makes the operation more straightforward. So you don't want to touch the fill system. So the septic piping and things like that need to be transferable between all the container sizes, and the size that's being filled doesn't really matter. So you can change the mold out and get smaller or larger containers, but you keep that flexibility, but you don't have to touch something that has to be revalidated once you adjust it. So you typically then size on the largest container, and then you can make, with limitations, you can make anything smaller than that.
So inter-product flexibility seems to be very good.
Yeah, yeah. If you're doing a family, if you're doing IVs, and you can do everything on one machine. So do all the sizes. If you're doing eye drops, you do everything that's available on that end.
Right, as long as it's the same formulation.
No. The changeover in that is very straightforward.
That's straightforward as well.
Yeah.
It just maybe I misunderstood.
Okay.
So let's talk about the delivery system then. So if I say one day I'm making drug A and the next day I'm going to make drug B, let's make it simple. They were both solutions.
Yeah.
Right. Sterile. So if I wanted to change over, what change parts do I need, or what do I got to do?
I mean, if it's both of them an eye drop and you're using the same container, there's nothing on the blow-fill seal machine. You push a button and you walk off. On your secondary packaging, you would have to do whatever, change the labels, change the boxes, whatever. But that's kind of practical. The machine cleans itself, steams itself, gets itself ready. You pre-program all the cleaning cycles into it. It takes whatever solutions it needs to do that. So it's
excellent,
automated in that respect.
So all upstream stuff is self-contained, CIP, the whole bit.
Correct.
Done.
And then you roll up a new tank, or you hook up a new tank, and then you run the next product. So
excellent. And typically, the tooling, that's pretty easy. That's a straightforward changeover. I mean,
within that family, as long as you're doing the similar sizes, it's 2 guys, a couple hours to change it over.
So where has been the uptake on this? I mean, have you seen it more in the CMO area, in the
the CMOs are taking over in the U.S.
You would think so, yeah. Seems to me like that's the way to go.
Yeah, the typical company that runs their own product doesn't fit a lot of people's business models anymore, and they don't want to do it themselves. There are still a few companies that want to do everything, but most are happy to give it to a professional outfit that does contract manufacturing.
So that's usually where you see your big sales, the guys who are doing contract packaging.
Correct.
Or contract manufacturing, right?
So they're the ones innovating into new areas, and then they get our equipment, and then we adapt it to what they need. So, and a little bit on the visual inspection side, this is a complement to do that, because one of the limitations for a blow-fill seal container or a hard to inspect container is that if you're going to go into that container and you move from glass, where you can use an automated system, and then you move to plastic where you have to use manual inspection because there's no automated system that can do it for you.
Well, let's talk a little bit about the automated thing.
So the big benefit there is that a limitation would be that if you have to use manual, it's a lot more expensive over the long run than an automated system. So the traditional glass systems, they take a glass vial, they spin it, stop it, and then as the water column spins, they take pictures and they find anything moving in there. And if they find something moving, we know you have particles. Now, what changes there is our ability to see through it and what particles are visible and things like that. So there's a lot of let's say, know-how and ability there. If you try to spin a plastic container like this, when it's in a block, you can't get it spinning, you can't get that water column. So what we developed was a technology, we actually vibrate it, we hold it on its side, we vibrate it kind of like your cell phone vibration, and then we take pictures from underneath, we rotate a station, and then we take pictures from above. Now we have our own special light, we can design the vibration, the harmonics for that particular container shape and the viscosity and things like that. But what we're looking for at first is any particles that sink, and by vibrating it, we see them move around, so we can do a comparison. When we look at the top, we see anything that floats or is in the top half of the container. And I guess what's different on our algorithm too is developed some new math behind it where we're adding and subtracting pictures, and it allows us to find things easier than the traditional, let's say, software behind it. So we don't really want to do the glass. The glass guys do a good job, and they're very high speed. What we are looking at is like blow-fill seal containers, flexible bags, things that the traditional systems don't do well at. And that's because, I have a bottle up here, so, spinning this would get you nowhere, or spinning a film bag like you see out there for an IV won't get you anything. But our technology, since we're just basically holding it and we're, imparting energy into the container, it allows us to really get into it. Compared to humans, we still have limitations because we have, optical, because the container's round and we have a meniscus. There are some areas that the cameras can't see into, or they're so distorted that we can't. But what we found is, when you're comparing against humans, they're not very good. And all the standard is we just have to beat the people at it. And even though we can probably, depending on a container, say this container, we can only see into 70% of it, not 100% of it. That means even if we do a perfect job, we're only at a 70% level because we can't look at the other 30%. We're still better than people at that because the people are still at a 45 or a 50% level. And this is, I think for me, the challenge, and also, the reality of it is that you really have to look at your process and bring it up, because the best optical systems still can't find everything. And you really only, there is a percent that are going to get by any system. It doesn't matter how good it is. None of them are perfect. People aren't perfect. Mechanical systems aren't perfect. Really, the only way to stop 100% stop contamination is not to have it there in the first place. So it's got to be a holistic look at the whole process. And again, it's back to at the beginning where we're talking, we can't really, inspect our way out of defects. So,
well, that's exactly the point. So let's talk here. So how would I inspect something like a bag, for example?
Yeah, the bags are a challenge too, particularly since a lot of them are pre-printed. So the films have come up. They've improved. They get thinner. They're multi-layered. They come pre-printed. So they have a lot of challenges, because if you try to look at something that's pre-printed, particularly if you have a traditional system where you're moving it, to get the computer to ignore the text is very difficult. And again, we're not perfect, but what we do is we'll ignore the printed side and we look through the unprinted side, and there we can look at for particle defects and, again, vibration systems. It's not as detailed as we can do with small containers, but it's still most likely better than what humans can do. And, on a step-by-step basis, we need to prove that out.
So if it's better than people, how far down does it go? Beyond visual, or?
No, nobody's, well, arguably, in the 100 micron range is practically subvisible for people.
Yeah.
Pretty much anything under 250 micron, people don't find regularly. Everybody's looking at particles that people normally can't find. And so we're finding, what we also know is there's plenty of particles below that level, even that machines can find. If you do your destructive testing, you can find particles in the 5 microns. Those are two levels where people are testing to. The FDA requirements, I think, for 25 micron are 3,040 containers per container. So people know there's particles in there. So
what about the variation in what you're looking for? Opacity, clarity. Not everything's going to be little
Correct.
Black particles. How do you deal with that?
We can inspect to the point where we can inspect. We can change light sources. We can, go to, non-visual light, and perhaps that allows us to see into the container where human eye can't see into it. There's a limit, though, at a certain point where, the machine vision can't see either. So but in general, those products you're going to have to do the best you can, and also probably do some type of sampling and destructive testing to back up your mechanical or human testing.
So let me ask you this then. So how do you challenge it? How do you validate it? How do you do a process qualification and a validation?
Sure. The basics are a NAP test. That's one method. You train people up. You do a risk analysis on your process. You create some samples that have particles that you could generate in your process, and then you feed those to people. So you sit the trained inspectors down, you test them on their sample defect set, and then you record how well they do, and you do that over weeks. And then you take that data and you take the same sample defect set and you run that through a machine, and then you compare them. So you're basically, ideally what you show is that the machine can do a better job than people can. There's a lot of, I think from a regulatory standpoint, if you're using an automated system, they like to see that, because trained inspectors that know that there are defects in there are better than your everyday inspector. If you have a defect rate of 1 in 2 million, that person could sit there for weeks and never see anything, and they get bored and they get lazy and they don't do a good job. Whereas the machine itself will do the same thing every time. So there's a big benefit to having an automated system.
Gotcha. So how do you challenge it? How do you challenge the system? Do you create? There's labs that do that. So you tell them what particles you can have, and then they will create a defect set, and they'll put particles into the containers, and then that's what you're looking for. And this is all part of, does
we
Rommelag set up the validation?
Yeah, we supply that services too. So yeah, we can do the NAP tests. So we have contract manufacturing, so we have trained inspectors, and so we can take containers and run them through our bank. Most customers want to run it through their own people if they have an existing facility.
Gotcha.
But if you don't have a facility and you're looking to build one, then definitely a way to do it would be through a contract side first. So.
Well, let me ask you this. If you're getting into it and this is brand new, why would you use people at all? Or is that just the way you got to do it? Just asking a stupid question.
Yeah, I think if you have a new product coming out right now, there's really no reason not to go automated.
I would think so, but again,
I think a little bit it's on that level, the production level. There's a threshold of cost versus, people, if you have a small runs and you don't run very often, there's a balance for the capital equipment and then the operations costs and things like that. If you have a continuous, if you're running every day and you have product going out all the time, then automation I would think would always be the best, economic sense.
Yeah.
If not, if you're the quality person, I would always want to see a machine doing it rather than people, but that there are economic reasons not to do it. So
I'm just a question. Is there other applications too? That would be, in other words, can we use this visual system to look at something else other than a blow fill seal? Or is it specific to that?
No. We're looking at bags. We're looking at IV type, large volume.
Yeah, I got that. Okay, yeah.
The next one, we knock off those two, and then the next one is we're going to look at anything that the traditional glass inspection systems don't do well. So maybe look at glass syringes, look at irregular products that are not, PET vials and things like that. Things that are not set up to, they're not glass, they're not cylindrical, they're not with a stopper in them that work in the traditional systems very well.
So if something was put up in a blister pack that one side was opaque, would this be an option for you or not? Or so you need visual, be able to see through both sides.
There's quality and there's, we can do a quality check on that, but I think for particle inspection it would be more difficult. So.
Yeah. Okay.
If you wanted to see everything was there and it didn't have any gross defects, or like in a blister, all the containers were there and one of them wasn't missing, then that we could do. So.
And again, getting back to what I had asked, if it was a segregated container, this is something you can confirm visually. In other words, if there was all the things are there.
Yep. Correct.
Okay.
Oh yeah.
No, the reason I asked is, I always say, in a former life, that was one of our biggest problems. We were only able to solve the problem by doing an isolation in the package. The problem was, well, how do you confirm all that? Well, next thing I knew, we had a line of people looking at it. Well,
yeah.
Eventually, it don't work. So let me ask you this about, the classic visual uses contrasting colors. Is this something that you bring into play here, or what?
We use infrared, so
infrared.
Yeah, that's the standard.
So it's not all visual light.
No, we're just using infrared light.
Gotcha.
So
All right.
that to us is the best for the plastic and,
Gotcha.
and seeing through it. That's not all fixed. It's just that's our development has gone in that direction. So it's better than visible light.
Good.
So
It's important to understand, because when we talk about comparing them to people, I think you got to level the playing field. And you got to say, okay, here's them to the circumstances what I'm going to look at.
Correct. They're using visible light. That's all they can use. So if the product is easier to inspect under other types of light, be it UV or infrared, then people are not good for that either.
Let me ask you this. I mean, one of the things I've seen with some of the more, because there's visual systems that'll do just about everything. I did a considerable amount of work in the device area, and they had one of the most involved optical inspection systems to assure the quality of the device was where it was supposed to be.
Yep.
And what they did was they got into a situation, where it was, well, what's a rejection, what's an acceptance, because you're going to see stuff you didn't see before.
Correct.
So how do you manage that? What guidance do you give people?
Yeah, we have we have a false reject rate. I mean, that's, ideally you find any real reject, but definitely you have false rejects. And that's something we work out with the customer. For example, we had a product that had, if we turned everything on and we looked the tightest we could, we had a false rejection rate of 6%, which is unacceptable. What we did is we actually derated our inspection in the computer, and we brought that down, and just as a number, we brought it down to about 95%. Our false reject went to well less than 1%. Now what we've traded off there is we are now theoretically not finding 5% of the defects. But since we are now comparing against people, in that particular case, the best person on one type of particle found 70% of those particles. So we are at a 95% level all the time, and they're at 70% at the best time. And so by derating our machine, we get rid of our false rejects, and we're still well above the required level. And that's particularly what you have to do. You have to actually, if you have a hard to inspect like foaming product, those can be very difficult, and because bubbles show up as particles. And so then you have to discriminate from them, or at least then do some mechanical manipulation of the container. We do an ultrasonic vibration to them to destroy the bubbles. And but each step like that you add, in general, your false reject rate goes up. So you have to really, you are compromising the system. You're telling it not to run at perfect levels because you don't want to find all these false rejects.
So does the ultrasound, when you apply it, create anything?
No, we tune it for the bubble,
Gotcha.
The product. I mean, it's variable, and you really do. It's a custom setup for that type of product. So, and most of those, they've got some surfactant in there, and a lot of it's from the handling upstream. The handling, getting it to the machine, the conveyor, whatever vibrates it. You can look at the things like that. I mean, if you don't create the bubbles to begin with, then you don't have to deal with them.
Yeah, well, that would seem to be a problem.
Yeah.
So this stuff is all tunable.
In general, yeah. I think the machine is 90% standard and 10% customized for every product. So you have some bells and whistles we can throw in, but you definitely need to sit there and test everything beforehand.
Okay, so we talked about blow fill seal, where it seems to be your sweet spot.
Yep.
Looks like there's a couple other opportunities. So is this the way, when you say custom, is this something I just bolt onto my system, or what do we do?
It's inline in the secondary packaging. So it'd be after the primary filling. So
Okay.
Be it blow fill seal or an IV bag
Gotcha.
or something like that, we would put that in line. You could do it offline too. So you could have it manually fed if you had the quantity wasn't that great, or maybe you have multiple systems and you only want one inspection machine, or you don't inspect every type of product, that can be done too.
Right.
So,
It would just seem to me if you're making the investment, it would be in line or at line.
yeah, inline
Right.
or outline, yeah, typically.
Okay. So it seems like an opportunity for a lot of folks to jump on board. But I was wondering, so what do you see as, I mean, we get into, do you see looking at defects in larger, one of the problems we get into is like recrystallization and stuff like that.
Yeah.
Do you see that as part of?
A little bit there. The products I've looked at, a lot of that happens after.
Seems to be time dependent,
after,
right?
Yeah, it happens. Somewhere between shipping the product and before use.
Gotcha.
Yeah, I mean, you could have one of these at the end user site at the hospital or something like that. But I mean, they're just looking at them, and those are normally larger particles, or product has changed colors, or
somebody messed up on the chemistry.
Correct.
That's usually what
yeah,
happens.
and normally we don't catch that stuff because it's not there when we
Right.
inspect.
Yep.
So
Yeah.
that's definitely a challenge. That's a huge deal out there, because I mean, you don't want to pass product out that looks fine and then somehow it changes.
I got to tell you, Tim, you'd be surprised how many guys formulate with a polymorph and they don't even realize that they're saturating the solution. But that's for another day. Tim, thank you very much.
Thank you.
Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.